And screen share. Okay. Can everyone see the PowerPoint, or do you see like presenter view? Can someone unmute and let me know?
See the PowerPoint.
Okay, great. Thanks, Tori. Awesome. Yeah, well, thanks everyone for being here today. I really appreciate you taking your time to do this quick training for this project. So, let's see. So the objectives for today. So I just wanted to give a quick overview of the project so far. So the first 5 to 10 minutes are going to be kind of just like an overview, just because I know we have a lot of new faces. There's a lot of returning faces, and I have talked to most of you already, but just sort of to make sure that everybody's on the same page about this project and everything we're doing. I just wanted to do a quick overview, so I'll be just about 5-10 minutes, and then we'll talk about the goals of this field season, this year's protocol, some of the logistics with getting everyone the gear at the right times, and then hopefully have time for questions and comments at the end. And so, yeah, I kind of gave that introduction of Harlequin Ducks. This is who we're studying. And so in the region, we have 4 long-term monitoring programs that have been going on. There's one in Glacier for many years, a couple decades. There's one in Glacier National Park on Upper McDonald Creek. There are 2 in Alberta, one in Banff National Park and one near Jasper National Park, and one in Idaho on the Lochsaw River. And all of these programs do yearly surveys, and all have been documenting declining numbers of breeding Harlequin ducks. Also, along with these breeding stream surveys, aerial surveys done of harlequins on their coasts have been documenting declining numbers of harlequin ducks on their wintering grounds. And so despite being incredibly tough, we've become increasingly more concerned about their status and their populations in this region. And so this is a figure showing all of the historical harlequin duck observations we have in the region. And this is a figure showing just observations in the last 15 years. And so as you can see, we have lost a lot of observations in many areas across the region. And so due to these documenting declining numbers of harlequins, apart from Alaska, they've been listed in each state and province. It's either critically imperiled, or vulnerable across their entire range. And so this is just a screenshot showing their status by province. And then historically, the way we've surveyed them here on their breeding grounds are with these ground-based foot surveys. And they rely on the direct in-person observations. And prior to this project, no one's ever really figured out what the detection probability of doing a single one of these foot surveys was. And we've been doing them for decades. And we've always thought that they're maybe not the most effective method. But the reasons that we've thought that is that there's a lot of conditions outside of the observer's control, such as flooding or drought, that can really make these the counts really highly variable, making the information they provide really highly variable. And so an example would be like spring flooding, which can result in harlequins moving into backwaters where they're unobservable. It can also affect human survey safety, which can affect detections when it's not safe for people to be in or near waterways. And then also, independent of just ducks moving and people not being able to see them, spring flooding could also affect occupancy if nests are being washed out and females leave the areas earlier than if their nests were successful. And so, as many of you know, they're just really challenging surveys to conduct, because you're either slipping and sliding your way up a stream or bushwhacking through miles of riparian brush. And so currently it's really impossible to know whether a lack of detection is related to bird abundance or human observer error. And then the other thing we learned a few years ago is that the genome of the Harlequin duck was assayed. So North Cascades had some extra funding, and their biologist, Jason Ransom, was interested in harlequins and went ahead and had this done with the National Genomics Lab of Wildlife and Fish Conservation that is in Missoula. And so thanks to him doing that, this opened up the door for us to consider using eDNA as a method for detecting Harlequin ducks. And I think most of you have some familiarity with eDNA, but just in case you don't, so DNA comes from anything that's left off an individual that contains cells. So this could be saliva, feces, feathers, blood. And then these cells are in the environment, and then we're taking environmental samples. So in our case, we're taking water samples to try and detect Harlequin duck DNA within the environment. And so working with many of you on this call, also many of you who are involved in the Harlequin Duck working group, it's become clear that there's an interest in better understanding, one, how our ground-based foot survey method works, and also to investigate if there's other better, more effective methods like eDNA. And so the point of all of this research is that region-wide, we do Harlequin duck surveys every year. However, we try and do an organized effort every 5 years, and the results of this study are to help inform this region-wide monitoring effort to hopefully help us gain a better understanding of Harlequin ducks and how they're doing in the region, and to also better understand what tools we have to detect Harlequin ducks, and to better understand all of the historic data and make it useful for current and future management. And so this leads us to the objectives, which are to investigate non-invasive detection strategies, which is what we've been doing the last 2 years, and then to predict Harlequin duck occupancy in the Northern Rockies, which is where we're headed with this field season and this next year of this project. And so I also, before, that's kind of like a very SparkNote introduction, but I wanted to give, just share some of the results from the last 2 years, just very quickly, another just like 5 minutes, just because I know a lot of you have been involved in this project. And I'm not sure how many of you have actually seen some of the results from all of the work we've done. So I just wanted to share some results real quick, and then we'll really dive into this year. And so over the last couple of years, we had 78 people conduct surveys for a total of 2,220 hours of surveys, so 92.5 collective days. We completed 118 ground-based foot surveys and collected 723 eDNA samples and placed just under 200 cameras and collected 2.5 million photos. And so these values that you're seeing are the average detection probability across all of the streams for a single replicate. So for example, this is just a single 5-kilometer ground-based foot survey and the average detection probability of doing one. And so for ground-based foot surveys, we found a 6% difference decrease between the 2 seasons and between single female and brood rearing seasons. And so during these 2 seasons, our detection probability is pretty similar, but a little bit of a decrease, which could just be that moms with babies are just a little bit more sensitive than single females, who seem to be they're still sensitive to observers, but a little bit less sensitive. And the main thing about this, and I want to point out about doing ground-based foot surveys, is that we've been doing these for a really long time, and we've just never known how effective this method is. But now we know that if there were ducks present on a stream, we really only had about a 50% chance of detecting them or not. And then the other thing we learned about ground-based foot surveys is that the more ducks there are on a stream, the higher the chances we are of seeing them. So yeah, as you would expect, the more ducks that people observed on their counts, just the higher the detection probability. And then the other thing that we learned is that when it comes to ground-based foot surveys, it turns out that observers really explain almost all of the variation within the detection, and a lot of the habitat variables were a lot less important in predicting detection than the observer team. And with that, we also learned that there's kind of the sweet spot right around the 5-kilometer, and asking people to survey 8 kilometers, which is close to 5 miles, was just too much because they had to go a lot farther with the same amount of time. And so people tended to move faster, and when they're moving faster, they're just not paying as much attention to the stream or not being as thorough. And so, as you would expect, the longer the distance, the harder the survey. So we kind of found that like 5 kilometers was the sweet spot for detecting Harlequin ducks or asking people to do these surveys. Moving on to eDNA, this is the average detection probability of detecting a duck given it's present on a stream, for taking just one single eDNA sample. And so eDNA, we found the largest difference between the two seasons. And kind of a hypothesis that I've thought about these results is that when we have single females, we tend to have, it might just be one bird, but they're a little bit more spread out evenly throughout a stream. However, when it comes to the brood mirroring season, a lot of the single females have maybe already left the area, or and so all of the DNAs in these broods that are kind of all in one location, and so the DNA might not be as free flowing throughout the stream as when they're more spread out. And what we found that affects DNA the most is the type of benthic substrate that the sample's taken in. So eDNA samples taken in boulder dominant areas have slightly higher detection probabilities. And this follows what other eDNA studies have found. And the reason for this is that benthic substrate actually absorbs DNA out of the water column. So fine sediment like sand and gravel will actually absorb DNA more quickly than larger rocks. And then we also used a spatial dependency test to learn about the best sampling scheme, and learned that between 750 and 1,000 meters, eDNA samples become independent of one another. And so we're not likely detecting the same ducks from there. And so with that, that kind of leads us to how we got to the sampling scheme that we're using this year of that 5 kilometers and having folks take eDNA samples every kilometer, which we'll get to just in a minute or two. And then for game cameras, this is, again, the mean detection probability of detecting a duck for just a single camera. And so for cameras, they were pretty similar, with only about a 3% difference between the two types of cameras. And we can clearly see that game cameras were the least effective method. However, sometimes they might still be the correct tool. And so an example that always comes to mind for me is on Upper McDonald Creek in Glacier. We have these beaver ponds that are really hard and dangerous for people to foot survey and take DNA, but maybe putting a camera there would be the right tool in that sort of situation. And so if we are going to use this tool for certain places and situations, it's important to understand how to use the tool. And so we did get some really cool photos, so I just wanted to share just a few highlights. This was a mountain lion, we had mountain goats, we had bears and other species of birds like the blue heron and wolves, and of course our favorite species, or my favorite species at least, Harlequin ducks. Yeah, and so kind of the main takeaways that we've learned so far about these game cameras is that time-lapse works just a little bit better than motion trigger because they're not relying on the bird actually detecting or triggering the camera, and then also placing cameras on calmer water, so pools and runs, was a little bit better than rapids and boulders and other ones. And then I've so far just talked about what the detection probability of doing a single foot survey, a single eDNA sample, or a single camera. But what's cool is the way that detection works is it's cumulative, so we can add the methods together to improve our detection. And so if we do a 5 kilometer foot survey, and then take eDNA every kilometer, we can increase our detection to close to 98%, and feel a lot more confident that we're detecting harlequins versus just doing single eDNA samples or a single foot survey. Yeah, and so this kind of leads me to the next objective, which is what this next season is all about. And so the focus of this year's is focused on the second part, which is working on predicting occupancy across the Northern Rockies. And so I showed this map at the beginning, but I wanted to revisit it. So this is all of the reported Harlequin duck observations with evidence of breeding from the last 15 years. And as we can see, there's a lot of spatial gaps within our information. And so in order to try and look at occupancy across the area, we really need to start filling in some of these gaps. And so we plan on doing this by using both eDNA and one more field season. And so eDNA filters, they can be analyzed for up to 8 species. And once a sample is taken, the genomics lab will store the filter, and we'll store the filters, and the information is actually cataloged in the eDNA atlas. And lucky for us, fish people in this region really like eDNA, especially those doing bull trout work. And so we've been able to reanalyze 230 fish samples for eDNA to start filling in some of those gaps. And so the orange dots on this map are the eDNA samples that we are able to have resampled to really start to fill in some of those gaps. And then this map is showing the full coverage of surveys, which I have talked to most everyone here about doing, eDNA, and then observations from the last 15 years that we will hopefully have all at the end of the seasons. And the different colors on this map are just different forest services, and that was for me to help me when I was making maps and keeping things clear of like who goes to what. So don't worry too much about those colors. But yeah, so ultimately the goal and reason for doing all of this is to produce a product kind of similar to this. So this is a figure from a paper showing predicted occupancy of rock bunting in Switzerland, where the darker the red, the more likely the area is to be occupied. And so we're working on building something like this, but for Harlequin ducks within the region that can hopefully be a good tool. And the goal, like I said, of doing all of this is just to try and identify important breeding areas for Harlequins, and maybe highlight potentially important areas that we're not looking to help us make decisions moving forward and where to put our time and our resources, and also to help with potential management decisions if that is needed. Okay, so let's talk about what this fieldwork season will look like and what you can expect from me. So I've already showed this figure, and so essentially this is what the protocol is. We are asking all of you to conduct a 5-kilometer ground-based foot survey and take eDNA samples every 1 kilometer. And so I think most of you have done one of these ground-based foot surveys, but just in case you can't, these are kind of the main points. So you will work in teams of at least 2 or more, and you walk upstream in the stream. And so walking upstream is important for this protocol because of the eDNA. We don't want to contaminate eDNA with our DNA by walking downstream and being above the samples. And so it's really important that we walk from downstream up, and then leapfrogging when necessary. So leapfrogging is what it sounds like. So like one person will stay on the stream if you can no longer walk in the stream or like directly adjacent to it. One person will stay there, and the other person on the team or other people will move ahead. And with this, you can just kind of use you can use whatever you want. If you guys use radios, you could use radios where the other person kind of moves ahead and gets to a spot where they can see up to or overlapping with where the person who is waiting in the river can see. And then the person who's waiting will then come up, meet that person, and then again leapfrog around. And so you could either use radios to like communicate where you say like, okay, I'm on the stream, you can come find me now. Or one of the things that I usually do is just use like a time where I say, okay, give me 3 minutes, and then it's up to the person who's leapfrogging around to make sure that they are back on the river, looking at the river for Harlequins in 3 minutes, and then the person behind will then move up and catch up to that person, and then give yourselves another 3 minutes or 5 minutes or however much time you think that you need to move around them. And so that's the main thing. And so you can use leapfrogging when, one, it's not safe to be in the water. Also, you should use leapfrogging if you notice that you are disturbing anything, like if you're disturbing Harlequins, or even sometimes I have pushed like American dippers where they just keep flying up and upstream. And so sometimes it's worth it to just do like one leapfrog just to get around that bird or that animal, just to stop disturbing them and pushing them upstream. And so that's essentially what the ground-based foot surveys are. And yeah, you just walk, and the most important thing is just one person always has their eyes on the stream. And that way that just ensures that if ducks swim or fly up or downstream, that they get noted so that you're not double counting birds if they did fly upstream and you spot them again, or you're not missing them if they go downstream. And so that's kind of the basics of ground-based foot surveys. In concept, pretty simple. In practice, they can be really challenging because there's a lot of bushwhacking involved. Yeah. And then as far as the data that we're collecting for ground-based foot surveys this year, they're essentially the same as last year and also the same as historic surveys. We just wanted to keep things similar as we have historically, just to help that data be helpful in comparing it to historic surveys. The main difference is the last 2 years the last 2 years I asked you all to measure stream flow at the beginning of each survey. This year, we're not asking for stream flow. And so once you get to your survey site, you can just take your first eDNA sample and start the survey. And this is what the data sheet looks like. So yeah, the time is just what time did you see a bird species. In Glacier National Park, we record all water bird species. But for Forest Service and FWP people, that's up to you what kind of information that your forest wants. So if your forest wants every species of water bird, record it, and that's great. If they don't, you can just record Harlequin ducks. That's up to you. Just maybe check in with your bios and see what information that you want, if you want all of that. And then yeah, so number of birds, how many, group type, and there's kind of a cheat sheet all up here. So females, broods, juveniles, chick class. And so there's also a cheat sheet up here. So class 1, downy, no feathers. They're like just born fluff balls. Class 2, partly feathered. And then class 3 are feathered and flightless. And so this is like class 3 is when the chicks look like the females, where it can be really difficult to tell the female from the chicks. But a trick for doing that, if you are seeing class 3 females, is to look for their legs, because the juveniles will have yellow legs still. And then yeah, where you saw them, latitude, longitude, geographic location. So this is like a bend after a cliff ban. And I think this is to just like help identify the area. Stream location, so like a pool, rapid, run, riffle, how far away you were when you saw them, awareness. So the awareness is the bird aware of you? And if the awareness changes, so say at first they are not aware of you, and then it does change, if you could add that to the comments, that would be really great, because the etho is their behavior. And so are they foraging, preening? And so we just want to know, like, if they do become aware of you and then all of a sudden their behavior changes, just note that in the comments. And then number of people on shore excluding yourself. So that's not including you. So this would be like if your stream is along a road or there's like a bunch of people fishing or something like that. If you could just note that, that would be great. And this, like I said, is all just sort of consistent with historic surveys, so just keeping this the same. Okay, eDNA, I'm going to share a video that the Genomics lab has made. Let me share this. Okay, so instead of me talking through eDNA, I figured I would let the experts.
I'm Kelly Kareem, research ecologist at the Aldo Leopold Wilderness Research Institute, a part of the U.S. Forest Service Rocky Mountain Research Station. This video demonstrates the collection of eDNA samples from streams following the Kareem et al. 2016 protocol developed at the U.S. Forest Service National Genomics Center for Wildlife and Fish Conservation. Stay tuned at the end of this video for information on how to contact us for support with your eDNA sampling and analysis needs. Here you see all of the materials necessary to collect an eDNA sample. When you work with the U.S. Forest Service National Genomics Center, All of the equipment and supplies may be provided to you for sample collection. Please note the hardcover carrying case is ideal for equipment storage, transporting the equipment in a vehicle, or when walking short distances from your vehicle to the sampling location. If you are working in the backcountry, you may prefer to leave the hardcover carrying case behind and carry the equipment in a backpack. Setting up. When you arrive at the sampling site, select a location with enough flat space to set up the pump and outflow bucket, and where you can safely reach, which could wash DNA from the equipment upstream, contaminating the sample. Step 1, connecting the pump to the battery. Connect the wiring to the back of the pump. Next, ensure that the connection between the wiring and the alligator clip adapter is secure. Attach the alligator clips to the battery, red clip to positive and black clip to negative. Step 2, installing the tubing. Pull the lever in the pump head to open it. Align the tubing and load the tubing into the pump head. Push the lever to close the pump head and ensure that the tubing is not being pinched along the edges. Place the outflow end of the tubing into the outflow bucket. You may find it helpful to secure the tubing to the side of the bucket, as the tubing may flop or jiggle when the pump motor is running. Step 3, testing the pump setup. To minimize the risk of contamination and ensure successful sampling, it is important to check the setup before taking any next steps. Turn on the pump. Next, ensure that the pump is providing suction. If the air is flowing out of the filter adapter, flip the forward reverse switch to change the direction of the pump. Once the pump is running properly, leave it turned on and move on to the next step. Step 4, opening the site kit. Open the gallon Ziploc bag containing sampling supplies and put on the gloves. To reduce the risk of contamination, work supplies out of the bag instead of reaching into the bag. Although gloves don't guarantee that a sample won't be contaminated, they do help reduce the risk of contamination. Step 5, preparing the forceps and sample bag. Retrieve the bag containing silica dust kit. Open the bag and set it aside in a safe location where you will be able to access it easily. Open the bag containing the sterile forceps and orient the forceps, keeping the tips inside the bag, but with the handle pointed outward for easy access. Step 6, connecting the filter cup to the hose. Open the bag containing the filter cup and rotate the cup so that the base is pointing outward. Leave the filter cup in the bag until you are ready to begin collecting the sample. Attach the hose to the filter cup by firmly pressing the adapter onto the base of the filter cup. Step 7, collecting the sample. Enter the stream and select a location where the water is flowing, preferably the foul way. To reduce the risk of contamination, avoid eddies and pools that could circulate DNA from clothing and equipment upstream. Keep your body and all equipment downstream of the sample site. Sample by dipping the filter cup repeatedly into the water or by submerging the filter cup and holding it in place. If the stream is shallow, you may weight the filter cup in place by securing rocks at the base of the filter cup. To minimize the risk of contamination, do not place your hands or any equipment upstream of the filter cup. Once the water is filtering, check to see if the bucket is filling with water. If the bucket is not filling, inspect the setup as in steps 2 and 3. Once the problem has been resolved, start the sampling process over with a new sterile site kit. It will take less than 10 minutes to pump a total of 5 liters of water. Occasionally, debris in the water can cause the filter to clog, prolonging this process. If this happens, stop filtering and preserve the current filter. Repeat the process using no more than 2 additional filters to pump a total of 5 liters of water. Step 8, draining and drying the sample. After filtering 5 liters of water, or less if the filter is clogging, remove the filter cup from the stream, keeping the pump on. Allow the remaining water filter cup. With the pump on, remove the filter cup by squeezing the sides of the cup near the base and popping it out. Step 10, folding the filter and securing the sample. Set the filter cup aside and grab the handle of the sterile forceps. Fold the filter into quarters. Add the filter to the bag with silica desiccant. Minimize the risk of contamination. Drop the filter into the bag, taking care not to insert your hands into the bag. Make sure the filter makes contact with the desiccant. Expel the air from the bag and seal it. You may now turn off the pump. Step 11, labeling the sample bag and envelope. Label the bag containing the sample and a paper envelope with the site information, including the site name, water body, date, GPS location, initials of the sample collector, and the number of liters pumped through the filter. Typically, you will have pumped 5 liters through a single filter. However, you may have used multiple filters to collect 5 liters. For example, you may have collected 5 liters by pumping 3 liters through one filter and 2 liters through a second filter. In this case, be sure the label reflects which filter represents 3 versus 2 liters. Step 12, placing the sample bag in the envelope. Seal the bag containing the sample in the envelope. 5 liters, you may place all individually preserved filters for that sample into the same envelope. Sample collection is now complete. Cleanup. Package the used filter cup and forceps to return for sterilization and reuse. Clean up any other materials from the site. On sample collection and troubleshooting, please see the
Okay. Awesome. Okay, so that was the eDNA, and I'll share that link. So if you're not doing your survey for another month or so, you can rewatch that and just get up to date. It's pretty straightforward. Everything that you can expect from me. If I'm sending you the pump, then you can expect all of the pieces that were shown in that presentation. If I'm just sending you the kits because you have a pump, you're probably more familiar with it and should have all of those, including the measuring device for measuring out 5 liters. One thing that you can do that I sometimes do on some of the more backcountry ones is I'll just bring an Nalgene and measure out 5 liters of water. Let's see. And so with the eDNA, you'll have to record all of the data 3 times, which I know can be a little annoying, but once is on the bag and once is on the sample envelope, like what they showed in the video, and both of those actually go to the genomics lab. And so I'm going to need you to record it once more on a data sheet that will come to me. And so this is what the data sheet looks like. Last year we took turbidity and a couple other measurements. This year we're just asking what the genomics lab needs and then benthic substrate and if you saw ducks or not. And so this should be a little bit more streamlined than it was the last couple of years where we're not using the Secchi discs anymore. So yeah, so this is the data all recorded. Benthic substrate, just which should be the predominant gravel type. So if it's sand or gravel, cobble or boulder, and then up at the top, you'll see like a little cheat sheet that gives you kind of like sizes of what we're asking for. And then ducks visible or not, just to kind of know if you could see ducks or not. Great. And so the last 2 years, just 1 second. Oh yeah, so the last 2 years we did a lot of bushwhacking, but after the first survey, we kind of knew what we could expect. This year, we're going into all new territory. And so therefore, when we were at the working group meeting, we talked about kind of a backup protocol if a stream was just absolutely unwalkable and surveyable. And so again, thanks to all of the data and work we did the last 2 years, we were able to learn that difference between the spatial sampling. And so that's why we came up with this backup protocol. That's if you cannot do the foot survey, we're asking you to take eDNA samples every 750 meters and go up to 6 kilometers, versus doing a 5 kilometer foot survey and taking eDNA samples every kilometer. And so instead of taking 6 samples, you would take 9 samples. And so if you think you're serving a stream that will be like this, please let me know ahead of time, so that I can make sure that you have enough filters. And then also I can update any maps that I'm going to send you. And so what you can expect from me is, so I have gone through for every stream that we have all talked about and discussed and measured out a 5 kilometer foot survey, because originally I had just sort of like loosely measured out streams and sent it to people to see if that would be a stream that would work for you or not. But now I have gone through for every single stream and I've measured out 5 kilometers, and I have made maps for everyone. And so you can expect those from me this week. I just finished making all those maps, so I will send them to you as both a georeference PDF if you use Avenza, and then I will also send them to you as a KMZ file if you use Gaia or Caltopo or Onyx or any of those other programs that you can upload into it. And essentially they look like, this is just a screenshot from GIS, but it looks like this, where there's a point every kilometer, to tell you when you're close to a kilometer and where to take your eDNA samples. And then I will also be sending everyone their eDNA equipment and kits. And then with that, I will also be including data sheets. And one other thing I forgot to mention with the eDNA is the last 2 years, I also sent people like a paint poll that was, you can extend the eDNA, and I will send those out again this year as well. So that way, if you can't stand in a stream and hold the cup down, you can clip the cup to this extended paint pole and reach it farther into the flow of the stream, essentially. So I will be sending those to everyone as well. Okay, so now I just wanted to take a minute and talk through kind of the schedules and gears and when I will be sending stuff to everybody, so that we're all on the same page, but also so everybody knows like when they can expect to get stuff from me. And so I'm gonna go kind of through this by forest/FWP region, just to like make it more clear. And so today I am planning on sending eDNA kits and a pump to the Bitterroot. And so my goal is to send everything at least 2 weeks before the week that you had put on the Microsoft Sheets, so that way it has plenty of time to get to you before your survey starts. And so today I am planning on sending for the Bitterroot your eDNA kits and pumps for the surveys of this week, July 15th. So for Big Creek, Whitecap Creek, and the Selway River. And then we do have limited eDNA pumps, so I am asking that within each forest you share the pump. And so then the next week, next Monday, I will send eDNA kits for Nez Perce and East Bitterroot. And so if you can just make sure that the pump gets over to the correct hands, that would be awesome. And then next, so FWP regions 1 and 2, Alyssa, I'm not sure if you're here or not. I know Tori is here. But I will just mail Big Creek's eDNA kits since I'm already going to send a package, because I know you are helping with that survey. But I will just mail that to them. But I would like to get you all of the other eDNA kits for all of the other creeks, and just on the Google Drive, I just, for my own scheduling purposes, put like the earliest date because some of you left comments of like it could be earliest this day. So I put the earliest date just to make sure that everyone had their kits on time. And so I will have those kits ready for you today as well. And so we can chat later and talk about if you want me to mail these, Tori, I can mail them to you as well. Or if we wanted to just meet somewhere, since I am in the Flathead, I could bring them to the FWP office or have meet you somewhere to hand these off. Okay, and then Kootenai National Forest. So I'll send eDNA kits for the West Fork of the Yak and the South Fork Big Creek with Alyssa, since she is helping with those. But next week I will mail the kits for Bull River, Vermilion River, and Cripple Horse Creek, as well as with a eDNA pump. I will make sure that those go out. Also, I don't have the date yet for Cripple Horse Creek. So if you can let me know when you're planning on doing that, that would be great so that I can make sure that you have all of the kits on time. Yeah. Oh, and then the other thing with the Kootenai National Forest, once you are done with Vermilion Creek this year, I am going to need to have you send the eDNA pump back as quickly as possible, just because we need to refurbish it, and send it with the Flathead National Forest. Because we're one pump short of having enough pumps for every group and forest. And so the Kootenai, the dates that you gave me for your surveys all end a couple weeks before the Flathead is starting their surveys. So if we can do a little pump handoff, that would be awesome. And then FWP region 3 and 4, so Owen, I will have all of your stuff ready by the 15th. And then also, this is a question also for Tori and Alyssa. We're also one pump short FWP, so I was wondering if there's any possibility of sharing the FWP pump with Owen, and getting it, because at least what I have on the calendar is the surveys that Tori and Alyssa are doing and Jesse are before the surveys that Owen's doing. It would be like we would have like a weekend, and so it might be tricky, but I'm not sure how much overlap you all have if you see each other, or like how easily it might be to do a pump handoff. But if we could maybe stay on afterwards to talk about this, that would be helpful, because that's just the last like pump handoff that we would just need to figure out. And then Gallatin National Forest, so I will send the 6 Mile eDNA kits with Owen, since I'm sending him all of his other kits, but I will be sending out all of your gear and pumps on the 15th for the surveys that you have scheduled those first few days of August. And then Helena Lewis and Clark, I didn't take a screenshot of yours. But the only difference is I'm gonna be sending you your gear 3 weeks in advance instead of 2 weeks in advance, because I know you are horse packing in, and you had asked if you could get your gear a little bit earlier. Let me know if you need your stuff earlier than the week of July 15th. But yeah, I have your gear sending out 3 weeks. Also, we can chat if you want to do a handoff like we've done the last couple years, or if you would prefer me to just mail the gear. And then Flathead, so like I said, we will try and get the pump back from the Kootenai and get that for you guys. And I'm not as concerned about getting you all the gear, just since I'm also up in the Flathead, so that should be an easy handoff. I can just, drive 10 minutes down the road to your office and get you everything. But I will have all of your gear ready for you by July 22nd. And for you guys, I'm just gonna send all of your kits at once. So even though your surveys are a couple weeks spread out, I'll make sure to get you all of that gear at once. Okay, and then what I still need is Casey, I'm not sure if you're on this call, but I just need your dates just to make sure that I get you your kits in time. And then I also need dates for 9 Mile and Hogback Creek. Again, both KCU and Idaho, and then I think Olin is on here for 9 Mile and Hogback. You guys both indicated that you have your own eDNA pumps. So I just have your kits and I just need to make sure that I can get it to you. And then I'm also gonna need mailing addresses and to talk to everyone about or like one person from each force about who to send their gear to and how to get their gear to them. And so that is, yeah, I think that is everything that I have for everyone today. So let's see, it's 10:18, so we have about 12 minutes. If people have questions, comments, concerns about gear, protocol, whatever, happy to answer any questions.
Hey, Holly, this is Casey with Fish and Game, Idaho Fish and Game.
Oh, hey, Casey.
Yeah, I think we'll probably do our surveys the last week of July, so like July 28th through August 3rd, something like that. So
Okay.
and I do have my own pump and stuff still, so I just need the DNA kits. So
Perfect. Okay. That is great. I will put that on my calendar and yeah, I'll do the same where I send you your stuff, like get it in the mail 2 weeks before that week so that I could see you with plenty of time.
okay, I'll throw my address in the chat too, just to make sure you still have that too. So
Perfect. Thanks, Casey.
Hey Holly,
Yeah.
since we have more than one person, I know that the eDNA video had just one person doing the whole thing. How do you typically like have the second or third person doing something with the pump to make it easier for the eDNA sampling?
Yeah, totally. So what we usually do is like when there's more people, one person just mans the pump. And so the person who's actually doing the kit isn't worrying about like turning it on and off. So someone else can man the pump. And also the tube that goes into the bucket that's filling up the 5 liters, that tube can be a little squirrely sometimes and like flop out of the bucket. So that person can just sort of like monitor that while one person just focuses on holding the pump or holding the sample in the water. And then also that person or the third person can start recording all of the data since you have to record that data 3 times. It's helpful to have someone like start doing that where the only one they can't do is the bag, the silicone bag until the filter's done, but they could fill out the data on the data sheet and on the envelope and kind of move that process along a little quicker.
Great, thanks.
Yeah.
Holly, Gerard here.
Hey, Gerard.
Thanks for the presentation. Triple your wages. Anyway, back to the foot survey. How do we know if some streams are not foot surveyable? I mean, yeah, how do you determine that? How do you know if you need 9 samples versus 5?
Yeah, so a lot of that is like, I know some people are familiar with some of the streams that we're going to already. So if they're just familiar because you're in that area, you work in that area and you think it might be that way, let me know. And if it's not, I will be sending 10 eDNA samples for every stream. So if you were to get to a stream and all of a sudden it's not sampleable or not walkable, you will have enough kits to do the shorter protocol or the like eDNA protocol. But if you know it's going to be not walkable, I'll just make sure to include a few more eDNA kits so that way you have more than one extra if a pump or if a sample, you end up needing multiple filters on a sample. So that's the main reason why I want to just know if you think, and then I can also update maps for people.
Okay, thank you.
Mhm.
Yeah, Holly, this is Ron Treadwell here in the Kootenai cabinet. And I had you shown they were going to mail kits for the Bull River and Vermilion River on July 24 and 25.
Yeah.
Now, those be two separate mailings then, or would you do one mailing that have the supplies for both creeks?
I will do, let me just double look at my calendar just to make sure I tell you correctly. Bull River, I'll do that in one mailing
Okay.
so that you just get all of that gear
And
at
so
once.
does the EDA kit have the pump as well, or are we providing the pump on that? And Chris is on vacation in Roslyn. He kind of talked about assisting with some of this. But I want to try
Yeah,
to,
so for those ones, I will provide a pump for you. So I'll
Okay.
include that.
everything
Yeah.
should come with those kits, and the I don't know, just some of the filters and stuff is the stuff that would be new with the kit, and the pump, you one pump for both kind of things, I'm sure, right?
Yeah.
Okay. And then had we kind of lined up to do the foot surveys the same time for bull and vermilion. I'm not quite the primary person on this one. I've been out of the loop a little bit on that one. Chris said, yeah, he was able to help out in some of that, but we're swamped with our staffing and what we can get done for our surveys. So I want to know what we'd kind of sort of semi-committed to you on that. But
Yeah, so for the Kootenai, you all committed for those streams, let me open this.
Vermilion and Bull.
Bull River and Vermilion Creek and Cripple Creek, and I don't have it open right at the moment, but I do have a list of like who committed to what. So if you wanted to stand for a minute after Ron, I can pull that up and tell you
All right, yeah, kind of
who committed to what.
Okay, thanks.
Yeah. And one thing that I forgot to mention, that Ron just reminded me of is after you have done all of the surveys, you can wait until your whole forest or FWP region is done with all of your surveys. But if you could send me back the eDNA kits, like completed the filters, sooner rather than later, that would be great, because we're gonna try and get them to the genomics lab to start being processed as soon as we can, to try and speed up the process of having them analyzed.
Ollie, would you like us here in Missoula to just submit those directly? I mean, I usually just drive over there and hand them straight to Jennifer.
Yeah, that would be great if you're in Missoula. That would be awesome.
Okay. And then we switched our week for doing Cache Creek, Trout Creek, and Big Creek in the Bitterroots to the week of the 22nd of July.
The 26th. Okay, awesome.
And then we can definitely get that eDNA pump to Region 3 right after that.
Okay. Awesome. That would be awesome if we could, yeah, get that handoff to Owen. And then that sounds good. I know I had on the schedule all of your surveys the same week, which I knew was probably not possible because there's 2 surveys a day and just a couple of you, but I wanted to make sure. So I'll still have all that gear ready for you, even though you're doing it the next week. I'll still have it ready for you today.
Okay. Sounds
Yeah.
great. Thank you.
What day was that handoff going to be, or week?
Ideally the handoff will be, because Owen, the dates that you gave me are your surveys on the week of August 29th. So that would be
Yep.
getting you that handoff that weekend. So sometime between the 26th and 28th, so that you would have them.
Okay.
We're traveling to Bozeman sometime between when we're doing our surveys here and when you would start yours, Owen. So I'll just bring it with
Oh,
me.
perfect. Sounds good.
Perfect. Great. Thanks, Tori and Owen. That helps a lot.
Yeah, you bet. Thanks for all the coordination. This is making it really easy.
Yeah, totally. Awesome. Is there any more comments or questions?
Yeah, Holly, Gerard again.
Hey, Gerard.
Do you prefer kneeling in the stream versus pole and clamp holding the cup?
I've always used the clamp holding the cup. Just the video shows her kneeling in the stream. I think you can use either method. The clamp is just, it's more or less the same thing, just like making sure you're holding it, within the stream. So yeah. Great.
Hey Holly, this is Tally from the Flathead.
Oh, hey,
I know
Tyler.
you talked to Katie Cagley now about serving the upper middle fork. And that's still something that we're able to do. So I was wondering if that's still kind of like a wish list thing for you. And if you'd like us to go ahead with that.
Oh, yeah, you might have emailed me that and I missed that. That would be great. We did keep enough samples, or order enough samples for those. So, let's talk about that. Maybe I'll send you and Katie and Jess a email separately to talk about that. But that would be awesome if we could get that done.
Okay, cool. That sounds good.
Sweet. Thanks, Tally. Great. All right. Well, thank you everyone for being here and for being part of this project. I really appreciate it. Yeah, keep your eye out for emails from me. I will send you an email once I mail the stuff. So mail your gear so that you know to keep an eye out for it. And then also I will be sending, like I said, this week, I made all of the maps for people and we'll send them your way. So I really appreciate you all being here.
Thank you, Holly.
Yeah.